So thank you for your kind words of introduction. And what I will do, although the title is fairly general, I have chosen to restrict myself to connections involving interneurons because I thought the topic of interneurons is anyway large enough to be part of such a session here. So, as I'm notoriously bad to acknowledge all my collaborators, I do this right at the beginning so that I don't forget this at the end. So for these works, I would like first of all to acknowledge Joachim Lübke, who has done a lot of the morphological work, especially some of the electron microscopic work. The same is also true for Jochen Steiger, who's also collaborated with me on the morphology here. Some two gifted PhD students, Moritz Helmstetter and Christian Körbel, have done some of the experimental work, and also initial work was done by a former PhD student of mine, Veronika Egger, who's heading a group in Munich, and everything was a lot of the work was done also in the Institute of Bert Sackmann, so I would like to acknowledge him here as well. The system we're working with is the rodent barrel cortex. The barrel cortex has in recent years become a very, very attractive model system because of its highly specific relationship between the sensory periphery and which is the whisker hair here, and the cortical structure. So what you see here is just one whisker hair that is connected via a sensory neuron in the trigeminal nerve to the trigeminal nucleus. From there you have a connection to the thalamus, and here a monosynaptic connection to a cortical barrel. In principle, you can say that one cortical barrel processes the information of one whisker hair, and the structure of the somatosensory cortex processing whisker information is basically a topographical representation of the whisker pad on the rodent snout. So this is basically what you can see here. You have here the cortical map, which is similar to the whisker pad on the rodent snout. It contains several cortical columns, which are particularly visible in layer 4 as so-called cortical barrels. The cortical barrels contain numerous synaptic connections, which you can identify in paired recordings as local circuits, and you can also study the properties of these synapses and eventually also the properties of the neuron, in which basically dendrites, soma, and axon communicate with one another. Here you can actually see what happens when a whisker hair is deflected. This here is the structure of a cortical barrel, and what you see here is basically the cortical activity measured as by a voltage-sensitive dye. You see it's very localized in this area, and it goes from an initial high subsequently to also this green here, which indicates that there is actually a depression right after the very high activity. We have studied in the last years numerous connections in the neocortex. These are all the excitatory connections, and this represents what is now called the canonical circuitry in sensory cortices. The information transfer is from layer 4 to layer 2-3, subsequently within layer 2-3, then to layer 5, and from layer 5 then to other cortical areas and subcortical structures. But this neglects completely the interneurons, and I would just like to show briefly, as by way of introduction, the heterogeneous morphology of interneurons. Just have a look at the green axons here. These are interneuron axons, and they can be highly local. They can be spread out like this, but also almost spindle-like, like this one. Again, we can, in layer 4, we have also very different types of axons. We have axons that go almost through a cortical column, but we have highly local axons that are, in principle, localized to a cortical barrel within layer 4. So, yes. So this will be the structure of my talk. I would like to talk to you about synaptic transmission involving interneurons in the sensory cortex, in particular the barrel cortex. And the major problem we are faced with is the fact that interneurons are notoriously difficult to classify. That is not just simply classify a cell as a cell type, or we don't know where they're projecting to, we don't know what they're doing, what their receptor complement is, and so on. So what I would like to do is I would like to show how we address this classification of interneurons. As an example here in layer 2-3 of rat barrel cortex, and show you that you can use the dendritic and axonal morphology of the sensory map here, the barrel column as a reference system. Then I would like to show you the properties of the excitatory synaptic transmission between layer 4 spiny neurons and identified interneurons. And subsequently, I would like to show you inhibitory synaptic transmission between fast spiking interneuron types and layer 4 spiny neurons. So in principle, the techniques I use will be paired recordings in patch clamp slice. So paired recordings here mean recordings from two different types of neurons that are synaptically coupled. You will functionally characterize these cells by evoking action potentials and recording the postsynaptic response in the coupled neuron. And as soon as when you have finished the functional characterization, you will do a morphological characterization, because we fill all these cells with biocytin, which allows us to recover the morphology of these cells and eventually also to identify synaptic contacts, which are here seen in blue. So we also perform a quantitative analysis of dendritic and axonal structure. I would just like to spend a few words on that. So what you can actually see here is how axons branch in a given area, and that they make contact with dendrites. The axon here is blue, the dendrite is in red. What that means is that although you don't know whether the axon makes a contact with a dendrite, it just tells you at least that this axon projects to a certain area. It does not project outside this area, so it has to make contact with neurons within that area. So in order to quantify that, so if you choose to have several neurons, what you do is you reconstruct the axonal structure and the dendritic structure, and here, what you see here, you put them on a grid system and calculate the axonal length within this grid and the dendritic length within this grid. Then this structure is actually smooth. This is just a smooth image of this here, and then this structure is smooth. And when you multiply these two structures, you actually, you would arrive at this structure. This is actually better seen. seen here. So you have here the dendrite, you multiply this and you arrive at this, which we call the predicted innervation domain, because this is the region where both neurons could make synaptic contacts. So from that you can actually calculate what we call density maps. This is an example from a publication we made. So here you have actually an image of a slice. Here you have several synaptic connections. So this was from 20 synaptic connections which were overlaid. And you arrive at this type of axonal density. You see this is a columnar axonal density. This is the dendritic density you arrive at, coming from here the white pyramidal cells. And a multiplication of this density with this density gives you here the innervation density, showing that in this example it is a columnar innervation density. So we try to use this type of procedure also with interneurons, but in addition we use the barrel column as a reference system. You may see, what you see here again is a neuron here in black with a green axon in the neocortex. You have here 3 cortical barrels. And the first parameter we chose was this here. This is the projection to deeper layers. So it's basically all the projections that are within this gray shaded area. Then we chose as a second factor the projection to adjacent columns, these gray shaded areas, and we defined them as, in this case, as laterality, and in this case as verticality of the axon. And if you now look at the structure, you see that neurons have fairly different projection patterns. Some neurons are completely lateral. They have virtually no projection to deeper layers, and some neurons are completely vertical. They project predominantly to deeper layers, but not very much locally. In order to classify that, because this is at this moment this is a qualitative classification. It's just something I see by eye. So we had to choose a quantitative measure, and what we did was we ran an unsupervised cluster analysis over our data. So in principle, we used just these 2 parameters coming from the cortical column system, and we arrived at 3 distinct groups of neurons. These different types of neurons can be seen here. The one type was we called a local inhibitor. The other one was a lateral inhibitor. And this one is a translaminar inhibitor, basically one that projects very strongly to deeper layers, to layer 5A and B, and sometimes also to layer 6. These are the density maps again. So these are the axon. Actually, with the density maps, you can see how local this is. It's basically within a cortical column. The lateral inhibitors project clearly to the 2 adjacent barrels, and the translaminar inhibitor is again very columnar, but projects to deeper layers. And from here, you could also see how the innervation would be for, say, pyramidal cells in layer 2-3 and spiny stellate cells in layer 4. So you see also that this innervation domain shows a strong innervation of the pyramidal cells within the barrel. This shows an innervation within adjacent barrels. This virtually no innervation or only a weak innervation in layer 2-3. If you compare that here to the innervation domains, you see that there's virtually no innervation here in layer 4 by these neurons, and a very weak one also by the lateral inhibitors, but a strong one by the translaminar inhibitors. So you can really classify these neurons also now by their projection patterns. However, things became more complicated when we delved a little bit deeper into the interneurons, because there are interneurons in layer 2, 3 that did not conform to the strategy, that the one's called chandelier cells. Chandelier cells are very peculiar. They exist both in hippocampus and in neocortex, and they seem to innervate the axon initial segment. What you can actually see with these chandelier cells, that they form these structures here, which are really the innervation points, the axon terminals on the axon initial segments. Then another type we found was a layer 1 inhibiting interneuron, an interneuron that had a very strong axonal domain in layer 1. We found 4 of those that are clearly different from all the other interneurons that fall here, that have virtually no axonal length in layer 1. So if you use these, if you come up with these cells as well, you see that these cells have, again, no innervation of spiny stellate cells. The layer 1 inhibitors innervate to some extent the layer 2, 3 pyramidal cells, but chandelier cells do so very little. And here you can actually see the axonal length distribution of these cells. This is another measure to quantify these cells, and you can see also how these layer 1 cells differ with respect to their length distribution within a layer. When we went, so because three classification parameters were not sufficient for us, we actually went on to use the dendritic structure of the cells as classification parameters, and here basically the insertion points of the dendrites. You can see here as next. This is a multipolar neuron, and this is a bipolar neuron which has a different structure where also the insertion points of the dendrites differ to a large extent. And what we tried to use here were dendritic parameters, meaning the total dendritic length, the length per dendrite, the number of primary dendrites, and their dendritic polarity. Then the axonal structure, as already described, and also, in addition, some measures of the action potential of the firing properties of the cells. So if we use the dendritic polarity, we come up with at least these 2 types. That is, one that is highly polar, like this one, and one that is virtually not polar. That is not polar at all. It shows if you use these polar histograms, you can almost see that it's a regular multipolar cell. The parameters we used here for the electrical analysis are, first of all, the appearance of the firing pattern, which is here a typical fast firing pattern, and here in this case a burst firing pattern with an initial burst and a strong adaptation later on. Here these 2 cells differ markedly with respect to their action potential. A very fast action potential with a half-width of 0.2 milliseconds, and a significantly broader action potential with about 0.4, 0.5 millisecond. And then you see the frequency adaptation that differs in these cells. There's little frequency adaptation for this cell, a marked frequency adaptation here, and also a difference in the AHP minimum. This shows virtually the same AHP, whereas in these cells the AHP actually decreases, as you can see here. However, we wondered whether the parameters, dendritic configuration and firing patterns were correlated, because we found that all the cells that showed this fast spiking pattern were actually multipolar type cells, and the cells that showed an initial burst and then a strong adaptation, these cells were the bipolar or bistratified neurons. And so what we did is we actually chose to make models of these cells using the original geometries of these cells, and incorporate sodium channels and some potassium channels in these cells at homogeneous densities. So it was a very crude model. But both cells had the same type of ion channels, and then we simulated the firing patterns of these cells. And we came up with this simulated firing pattern for this cell type and this simulated firing pattern for this cell type. And although there is some difference to the actually measured firing pattern, you can see that this is much similar to this one, and you have an initial burst here for this one, which you would never see in these cells. This means actually that dendritic and electrophysiological parameters are highly correlated. So I would like to come to this point which you addressed. This is basically how can we classify how can we classify a neuron, and which is the most important parameter for classifying cells. And what we did here is we used the different parameters we had, so the electrophysiological, the dendritic morphology, and the axonal morphology, and reduced their respective weights. Starting with this point here. Here all have the same weight, and you can see here that using all these parameters, cells fall in 1, 2, 3, 4, 5, 6, 7, 8, 9 groups. Now we gradually reduce the weight until the axon is no longer used. So this is 0 here. And what you arrive at here is a completely scrambled classification. So the axon is clearly an extremely important parameter for the classification, because it classifies not only the structure of the cell but also the projection pattern, the innervation pattern of this cell. If you now use the dendrite and you reduce the weight of the dendritic parameters, you can see here the original point, and you can see here the end. You can still see that there are groups. So you lose a little bit of information, but not terribly much. The same is actually true here. You still maintain the groups. Some of the groups are no longer in existence, but there's still 1, 2, 3, 4, 5 groups of R9. So the most important parameter is here the axon, because the axon actually, if you remove the axonal geometry, you get no proper classification at all. So the previous classifications, which were based on electrophysiology and dendritic geometry, are not were not really sensible. And I will show you another example where we found that to be the case. So these are basically the interneurons we arrived at. I'll just show a few. These interneurons from this group, local interneurons from this group, which are more spindle-like. Then these lateral interneurons, different types, and also translaminar interneurons. And the final outcome here was basically to classify interneurons in 4 different local groups, 3 different lateral groups, and 2 translaminar groups. And then we have also the chandelier cells and the lateral inhibitors, the layer 1 inhibitors, and we have also come up with some comparison with data from other studies. Basically, what are the local interneurons here? We see that our local interneurons are probably similar to the small basket cells. The local 2 are similar to neurogliaform cells. And then we have also the bitufted cells. But the bitufted geometry is not a very good classifier. The lateral inhibitors are likely to be large basket cells, or the non-fast spiking basket cells that have been previously published, or tripolar cells. And translaminar cells are also these bitufted cells, but with a much longer axon. And then we have these layer 1 inhibitors, which are likely to be Martinotti cells. So we now used our terminology by looking at synaptic connections between layer 4 spiny neurons and these interneurons. This is just shown here, a barrel cortex slice, one pipette here on a spiny stellate, and one in layer 2-3. These are the two cell types you can see. And this is the dendritic geometry of the spiny stellate and of the interneuron. And in blue, it's probably, if the light could be switched off, you can see it better, unfortunately. So in blue is the axon of the spiny stellate that innervates the dendrite of the interneuron. These are paired recordings. You see here the action potential in the spiny stellate innervating EPSPs in the interneuron. The latency is very narrow, so it's about 2.5 milliseconds, and the synaptic efficacy is small with less than millivolt in amplitude. There is only a very weak relationship between latency and PSP amplitude. Yes, what you can also see is that there is that these cells exhibit a relatively high failure rate when the PSP amplitude is small, and only when the PSP amplitude exceeds 3, you get actually very little failure. We found, we looked at different interneurons types. This is a local inhibitor where we stimulated the cell with 3 pulses, and what you can see here is that the cell actually shows a paired pulse depression and a pronounced one. So here are actually the histograms from this mean amplitude went to this, and the third one had this mean amplitude, so it's a clear-cut depression. This cell was different. So this is actually a translaminar cell, and here you find a paired pulse facilitation. So you have a very weak PSP to begin with, but the following PSPs are much larger. This actually indicates that the classification of the cell types is important when you want to understand synaptic parameters, because invariably these cell types here showed a facilitation, whereas the local inhibitor cell I showed before showed a synaptic depression. So we came up with, we looked at several cell types, and we found some cells that showed a paired pulse depression here, where some cells that showed a paired pulse facilitation, whereas the majority of neurons showed paired pulse depression. So at at this cell we were actually able to identify the synaptic contacts, and it's very important to note that these connection here, this is a layer 4 to layer 2-3 interneuron connection, has very few synaptic contacts. Here in blue you can actually see the synaptic contacts made by this neuron on the dendrites of this neuron. I would also like to point out that these are dendritic inhibitory contacts and not very proximal ones. They are actually 100 microns from the soma. And we were also able to verify these contacts by electron microscopy. So you can actually see here now how these local inhibitor cells that were the depressing ones innervate the layer 2-3 pyramidal cells. These are all recordings from pairs we did. And the same is here for lateral inhibitors and for some translaminar connections as well. So it's basically here, as a group, I would like to point out these are the differences we find for this lateral, for this local connection here. We find a high failure rate and facilitating paired pulse ratio. For these two other types, which were the most frequent, we actually found, and where we could do statistics on, we found depressing connections and relatively low failure rates. So basically, the sequence of activation we can now show is the following. We have activation of the spiny stellate cell that in turn activates an interneuron and can subsequently inhibit a pyramidal cell. Sorry. Yes, I would. So I would like then turn to some inhibitory contacts, and I find this type of neuron especially peculiar because it's a neuron within layer 4, and as you can see from its morphology, it's highly compact. It has an axon that is actually remains within a barrel. We found some different types of interneurons in layer 4 as well, some that projected outside the barrel, but some, like these, and these, that remain really very strictly within a cortical barrel. And as you can see here, a little bit better. And we were therefore interested in looking at the properties of this particular type of cell. And again, we had to go through a longish procedure to identify these cells, and we used the axon again, but in this case, we used two different parameters. You can actually see that again. First of all, what fraction of the axon remains within the barrel, and what fraction of the axon remains within the home column. So this is basically what we call axon-barrel ratio. So axon in the barrel and axon in the home column. You can see also this distribution here and a particular type of cells that have an axon that's virtually exclusively within the barrel. Oops. And we were able to group these cells, and our initial grouping was confirmed by the cluster analysis, and the cluster analysis shows clearly that you have a cell type that has a low axon home column ratio, so a low axon barrel ratio, and two other cell types that are intermediate and on the other hand have an extremely high axon barrel ratio. For these cell types here, and this is what I will focus on, we have an axon barrel ratio of about 90%. You can see here the axon density plots. You see that the axon remains completely within the barrel. But when you now look at these cells, and this is what we find very important for classification of cells, you probably have heard about classification of interneurons as being fast spiking. The problem here is that these cells have all different axonal geometries, and you can actually see that they have it. They fall in different clusters. But if you look at the firing pattern of these cells, both at low and high resolution, they're almost identical. If you would run a statistical analysis over this firing pattern, you wouldn't be able to find a difference. And I can show you that in this table. These are the three different clusters. For example, look at the maximum firing frequency. That is virtually identical. Look at the thresholds. These are identical. The half-width of these action potentials are identical. So you have cells that have, if I would like to remind you, cells that have completely different geometries. Look, this one projects to layer 2, 3, and also down here, and the other one remains within layer 4. So although these cells are different, they have different firing patterns. They don't have different firing patterns. So yes, so now we have decided to use just the axonal geometry as the defining criterion, and the major criterion here is for us the 89% of the axon in the barrel. So the firing pattern itself is a very poor predictor of the axonal projection pattern. If you look at these different types of interneurons, so they are now color coded. You find that they are all over the place. Here, there's no clear relationship between firing pattern, in this case, maximum firing frequency and half width, or adaptation ratio and maximum firing frequency. So it is now very important to know what these cells are look like, and to have at least a proper idea about their axonal geometry. So what we did then is we looked at the synaptic parameters of this particular barrel inhibitor interneuron with a high axon barrel ratio. So I'm talking about the properties of these cells now. And we see here the PSPs. You see also their slow waveforms. You see that they have a relatively high CV compared to, for example, spiny stellate connections by themselves. So it's around 0.5. And you also see that these connections are not extremely efficacious. And again, we did some paired pulse behavior. We saw that this particular type of connection is clearly depressing.
Sorry, what's your chloride driving force?
The chloride driving force was, so we used 4 chloride in the pipette. So we had a chloride reversal of about -80. Yeah. But these recordings were all done at -55, because otherwise the signal would have been too small, because the resting potential of these cells is around -75. So here again is the connected cell with the variation of the PSPs, and what we found were 2 types of connections. We found, first of all, one connection where we had only an inhibitory connection but no reciprocal connection, as you can see here. You find action potential in the interneuron and get a IPSP. But if you find action potential in the spiny stellate, the interneuron does not respond. But we also had reciprocal connections and a very high fraction where we could fire, again here, the interneuron and see an IPSP, but on firing the spiny stellate, we saw an EPSP in the interneuron. Actually, the connectivity of these cells is extremely high. So if you've ever done paired recordings on synaptically coupled cells, you'll normally find that the connectivity is in the range of 10% or much, much lower even. Here you have a connectivity ratio that is about 60%, and of those 60% of cells, 70 were reciprocally connected. So we always call that a patch's paradise, because you don't have to search for synaptic connections, which is often what you have to do for excitatory connections. In contrast to the spiny stellate cells connection, we also found that the latency of this connection is very brief. So you have, on firing the action potential in the interneuron, you have a IPSP at a latency of 0.5 millisecond. So if you compare that to a spiny stellate connection, that's about half of the value in a spiny stellate. So this is a summary of. This is a summary of the data I've presented you. So we used always -55 in the interneurons as in the spiny stellates as holding potential, and -70 approximately in the interneuron as holding potential. And the amplitude we find here is 1 millivolt, but actually at the true resting potential, the amplitude is significantly smaller. And the EPSP amplitude here is relatively large. You see also that the CV, which is an indicator of the release probability of these cells, is higher for the IPSPs than for EPSPs, as you can also see by the different failure rates. The failure rate is up by 200% here. And here again, the latency between these two cell types differ by a factor of 2. So again, this is with this I just want to show the innervation domains of these 3 different cell types, and you should concentrate on the lower one, which is the cell type I was actually discussing here. You see this highly local innervation of these cells, so it's basically a cell type that probably innervates only one and inhibits only one particular type of neuron, namely the spiny stellate neuron in layer 4. So, and this is this should show you the parameters. So these are the different cell types. We did a few recordings of the others, but these clusters 3 cell types differ markedly from in amplitude from these, and were cells with a relatively low reliability, but a very fast inhibition. We also looked here at the synaptic contacts, and this is an example of a reciprocal connected cell. So this cell had 3 inhibitory contacts on the spiny stellate cells and 2 excitatory contacts on the interneuron. We used here also statistics, namely in that we looked here at 11 cells and here 6 cells, and what we found is here the filled bars are from our barrel inhibitor cells. You see that the number of contacts never exceeded 6, so it's a fairly low number of contacts we find, and these contacts are from very proximal to extremely distal. The excitatory, however, the distribution of the GABAergic synapses does not differ very much from the distribution of excitatory synapses. Excitatory synapses can be as proximal as GABAergic contacts can be. The number of excitatory synapses on the GABAergic neuron are, in this case, clearly lower than the GABAergic synapses. So what you see here is basically the initial inhibition that occurs via feed-forward excitation of the inhibitory interneuron and the spiny stellate through the thalamus. So you activate the spiny stellate initially, and via a disynaptic circuit, you inhibit the spiny stellate again, and this inhibition occurs very quickly. So you can see here the summed signal. This is the PSP in the spiny stellate that's inhibited within half a millisecond, so that the PSP is actually already reduced, and this results in a very brief activation window for spiny stellate cells, and it's probably also the reason why in vivo you can never measure more than one action potential in one spiny stellate neuron upon sensory activation. It may be the reason for what we now call the sparse sensory coding in sensory cortices, which actually shows that the activation of neurons is between 10 and 25%, and that always one action potential occurs upon sensory activation. So what I've shown you here is that spiny stellate cells are innervated by 3 different types of fast-spiking interneurons, which differ profoundly in their axonal projection pattern, and that the most distinct type of those is the neuron which has a very local axonal domain within a cortical barrel. The connectivity between the spiny stellate cells and these interneurons is extremely high, 67%, and of those are 69 reciprocally connected. The average IPSP at minus 55 millivolts is 0.9 millivolt, and the latency is extremely short, with 0.5 millisecond and a rise time of 1.3 millisecond. The connection is less reliable than that of excitatory connections in layer 4. However, where the CV is 0.37, and the CV of these IPSPs is almost 0.5. The connection between these barrel inhibitor or cluster 3 interneurons and the spiny stellate cells are very effective feed-forward inhibiting microcircuit in the neocortex, and are probably in the first position to inhibit sensory input to the neocortex. Thank you.